bioinformatics micromodules, Markov models, and hidden Markov models. In this module, we discuss the task of annotating biological sequences and introduce the concepts of Markov models and hidden Markov models, which are commonly used in this annotation task. Recall that many types of biological objects can be represented by a linear sequence. For example, DNA is a sequence of nucleotides. RNA is also a sequence of nucleotides, but it contains urosols instead of thymines. And protein is a sequence of amino acids. For each type of sequences, we want to perform annotation, which is to record the meaning of certain parts of the sequences. For example, on the DNA, there are places that can be transcribed to produce RNA products, which we call genes. And thus, a first level of DNA sequence annotation is to record the starting and ending positions of each gene. Besides genes, we also want to locate different types of regulatory elements, such as promoters, enhancers, and insulators. Similarly, for RNA sequences, we want to determine the locations of exons, splice sites, and introns, which will determine the sequence of the processed RNA. Then for messenger RNAs, we also want to find out the locations of the 5' and 3' untranslated regions and the coding sequence, which will determine the protein to be encoded. As for proteins, usually we are interested in locating the domains, which are functional units that serve different purposes. There are also other types of biological objects that can be represented by a linear sequence, as well as other types of annotations, which we do not discuss here. Let's see a toy example to get some feelings about the annotation process. Suppose you are given this DNA sequence. It is a short sequence with less than 300 nucleotides, but it can already give us some ideas about the difficulties that we would face when we are annotating the whole human genome, which has 3 billion nucleotides in each haploid copy. The annotation task that we want to perform here is gene finding, which basically asks whether there is a gene in this sequence, and if so, where it is. There are many ways to find genes. For example, if we have performed a molecular experiment to find out the sequence of an RNA transcript, we can align it back to the genomic sequence to find out where it comes from. Here, we take a different approach that is based on statistical modeling. In a nutshell, this approach considers different candidate subsequences and uses properties of genes to score each candidate, where a higher score means that the subsequence is more likely a gene. For example, suppose genes usually have a low percentage of A's. Then this highlighted subsequence may receive a high gene candidate score since it contains only C's, G's, and T's. Of course, this property about a low percentage of A's is just a fake property that we made up, and it is not really true for real genes. In actual gene finding, our first goal is to find out the real properties of genes. After that, we decide how subsequences should be scored based on these properties. And finally, we need algorithms that can compare the scores of many different subsequences efficiently. To achieve these 3 goals, we need to first introduce some key concepts. First, we define a concept called state to mean each functional category that we are considering. In the basic gene finding task, there are only 2 states, namely genic regions, which are regions within a gene, and endogenic regions, which are regions not within a gene. Based on this concept, we can redefine the gene finding task as determining the state of each nucleotide in the DNA sequence. At the beginning, when we are given only this sequence, the actual state of each nucleotide is not observable, or in other words, it is hidden. In order to infer the hidden states, we make use of some observations. In our gene finding example, the observations are the nucleotides in the DNA sequence. Finally, another important concept is that successive states have dependencies rather than being independent of each other. For example, since each human protein coding gene is several thousand nucleotides long on average, we will expect if a nucleotide is within a gene, the next nucleotide is also likely within the same gene. In other words, the state of a nucleotide depends to some extent on the state of the previous nucleotide. We can make this dependency assumption formal by using a Markov model, named after the Russian mathematician Andrei Markov. In a Markov model, the current state depends on the previous k states, where k is a parameter that determines the extent of dependency. If k equals 1, we call the model a first-order Markov model. If k equals 2, we call it a second-order Markov model, and so on. So what do we exactly mean when we say the current state depends on the previous k state? We mean that the probability of the state at position t, which we denote as qt, given all previous states, is equal to the probability of it given only the previous k states. In other words, given the previous k states, the current state is independent of all the other previous states. In addition to this state dependency assumption, in a Markov model, we also assume that the current observation depends only on the current state. Formally, it means that the probability of the observation at position t, which we denote as OT, given the current state and all previous states and observations, is equal to the probability of it given the current state alone. To understand the Markov model better, let's consider a coin flipping game, which is described by a first-order Markov model. Suppose there are two coins, A and B, where coin A is a fair coin, which means if you flip it many times, you expect half the times you would get head and half the times you would get tail. Coin B, on the other hand, is an unfair coin, which has a 25% chance of head and 75% chance of tail. Now suppose we play the coin flipping game as follows. First, I randomly choose one of the two coins and put the other coin into a pocket. I flip the coin chosen and observe the outcome, and then put the coin also to the pocket. I then repeat the process by drawing a coin out from the pocket and toss it. However, the coin that I draw each time depends on the coin drawn last time, because the last coin has a higher chance of staying on top in the pocket. Also, suppose coin B is a heavier coin, and thus it usually sinks to the bottom of the bucket, making it less likely to be drawn in general. Considering these two factors together, we have this transition probability table that if coin A is used this time, next time there is a 90% chance that it will be used again, and a 10% chance that coin B will be used instead. On the other hand, if coin B is used this time, next time there is an 80% chance that coin A will be used and a 20% chance that coin B will be used. We can summarize the whole process by a graphical representation. Here we use the two nodes labeled A and B to represent the two coins. The left two arrows pointing to these nodes indicate the initial probabilities of choosing the coins. The arrows pointing from one node to the other or back to itself indicate the probabilities of drawing a coin next time given the coin drawn this time. Finally, the thick arrows on the right indicate the probabilities of the outcomes given the coin used. Given this setup, one possible run of the game is as follows. First, we randomly draw a coin, and it turns out to be coin B. We flip it and get a tail. Next, we put the coin back to the pocket and then draw a coin out from the pocket, which turns out to be coin A. We flip the coin and we get a head. Again, we put the coin back to the pocket and then draw a coin out of it, which turns out to be coin A. We flip it and we get a tail. In this example run of the Markov model, we assume we know which coin is used each time. But if the two coins look exactly the same, we will not actually know which coin is used. In that case, we can only see the outcomes of the coin flips, but cannot know which coin is used each time. Since the states, that is, the coins used, are hidden, this is called a hidden Markov model. Now, let's return to our original gene finding task to see how the coin flipping game is related to it. In the coin flipping game, there are two states, which correspond to the two coins. In the gene finding task, we also have two states, which correspond to genic and indigenic positions. Regarding the observations, each coin flip produces either a head or a tail, while in the DNA sequence, the nucleotides are the observations, and we assume genic regions and indigenic regions have different distributions of nucleotides, just like the two coins having different distributions of head and tail. Finally, we should note that although in the coin flipping game, each step is a time point, while in the gene finding task, each step is a nucleotide position, we can generally consider them both as a sequence of events without worrying about the meaning of each step. By now, we have discussed how we can systematically record properties of different states using Markov models, which was our original first goal. In the next module, we will study the second goal, namely scoring each subsequence based on statistical properties of the states.